Hello, everyone. I'm Sharon Crook, and I'm delighted to be here for session 3 of the INCF Assembly. Our topic is Fair Approaches for Computational Neuroscience. I'm really looking forward to the talks today, and I want to thank the INCF for including this topic. I think it's very timely and important. Before we get started with the talks, I'm going to give a very brief introduction to the topic. So I will give an overview of FAIR principles and how I think they relate to computational neuroscience. We'll see that some themes arise around the FAIR principles in this domain, and I'll give a few examples of resources that support FAIR approaches for computational neuroscience. So I think everyone is probably familiar with the Wilkinson et al. 2016 paper on the FAIR guiding principles for data. And you likely know that FAIR stands for findability, accessibility, interoperability, and reusability. These are the main 4 principles. Within the paper, the authors also outline requirements that they think are important for supporting each of these areas. And so I want to discuss these 4 areas and the requirements in each area and how I think they might relate to models. So of course, we think of models as being informed by data, whether we're doing modeling that is extremely data-driven or more phenomenological. We still compare the output from models to data. We use models to predict what we might see in new experiments and drive neuroscience forward. And I like to think of modeling as a different form of data analysis. We use models to understand data, but also to hopefully better understand the biological processes that created those data. So if we focus on findability, then the underlying requirements that are outlined that maybe are the most relevant here are that we need rich metadata associated with data that are needed for automatic discovery. We need unique and persistent identifiers for data so that we can find them, and we can find them now and can find them into the future. And of course, we need searchable resources where data can be shared and data can be selected for reuse. And these same requirements, of course, apply to models. It's very important that we strongly associate metadata with models, that they have these unique identifiers, and that we have searchable resources for sharing and selecting models. And luckily, there are a number of groups that have been working on platforms, databases in support of this idea for quite some time. There are just a few examples here. And of course, we need to be sure that these resources go across scales, from the scale of molecular interactions all the way up to whole brains. In support of metadata, we need vocabularies that we can use, preferably ontologies, so that we have agreement on what terms mean. And luckily, again, there are resources that exist that have been designed to help us with these aspects. So for example, Interlex from NIF, the Neuroscience Information Framework, provides a federated vocabulary that relies on existing ontologies. All right, moving on to accessibility. The main requirements outlined in the paper that are relevant here are that data should be open and free. And of course, this also applies to models. So luckily, when we publish models, we think of them as open and free. People are free to re-implement them, to reuse them. But in many cases, practically, a publication isn't enough for that. We need code to be shared, or even better, we need some sort of description of the model that we can use to access all aspects of the model. And I would also like to mention that I believe institutions like the INCF that are constantly advocating for open and free data and models have been really important for sort of changing the mindset of our community towards accessibility of models. All right, moving on to interoperability. The most important requirements are that we need standardized knowledge representations for data and vocabularies. So again, here's this aspect of ontologies being important, you know, this standardized way of representing knowledge. And then we also need standards for representing for represented models. And again, luckily, a number of groups have been working on things like this for quite some time. And here are just a few examples. Whether you're looking at standards that are more descriptive or standards that are procedural, these standardized formats are extremely important for interoperability of models. We want to be able to, you know, take the same model and use different resources for developing models, different software for developing, for simulating, for validating, for sharing models, for visualizing models, or model outputs. And so these standards are really important for interoperability. And again, I want to mention that another thing that's very important is having organizations like the INCF that have a rigorous process for endorsing these standards. And finally, when we think about reusability, we're going to see some of the same themes. The requirements that come up in the publication are we need accessible usage licenses, we need detailed provenance for data, and we need community standards again. And I would add that for models, not only do we need community standards, but we need software to support those standards. So similar to interoperability, we just don't have reusability of models without the software support for these standards. And so, you know, returning to this original idea of how models interact with data and thinking about the FAIR principles that I've just gone through, some of the themes that come up are obviously that we need metadata and we need ontologies to support FAIR approaches. We have resources like Interlex for doing this. We need open source resources for sharing models and selecting them for reuse, which we have. Again, these are only some examples here. This is not an exhaustive list of resources. We need standardized formats for models at different scales, from the molecular to network and beyond, whether declarative or procedural. And we also need standardized formats, of course, for data. And then lastly, we need software support for all these standards, whether it's simulations or maybe software for validating models against data. And we'll hear about some of these things today. Okay, so that's basically it. For those of you at home, you can view this later. And here are the resources that I mentioned today. Thank you very much.
Thank you for joining me today. I am going to be speaking about my experiences with FAIR in our work on creating data-driven, personalized, large-scale dynamic network models. And I will start by describing the various types of data that we use to tailor our models and give you an example of our workflow. I will then discuss some of the challenges to FAIR that we face that I believe are specific to these types of models, and I will talk about some of the FAIR practices we've built into our workflow. Then I will speak to how we are able to continue to improve on these practices by scaling up and moving our work into the cloud. And finally, I'll finish with a discussion on some of the remaining challenges to FAIR that we still face when working with these personalized models. With the growing number of large data sharing initiatives, there's an incredible opportunity for building large and heterogeneous sets of personalized models. And I've listed a few examples here, such as the Human Connect Home project or the UK Biobank data sets. But there are many, many more that are either currently available or soon to be available. The last three I've listed here, the ADNI for Alzheimer's disease, the PPMI for Parkinson's disease, and the CAN-CAN data set of aging subjects are the ones that I am most familiar with and the ones that we are currently modeling and from which I've drawn my particular perspective with respect to FAIR practices. And what we're doing with these data sets is using the virtual brain platform to create large-scale models of network dynamics and that are personalized to an individual using their own neuroimaging data. And this can be in the form of any number of neuroimaging modalities. Here's an example of a typical workflow using MRI data. So where the T1 weighted image and the diffusion MRI data are used to map the subject's structural connectome. Each node in our network is represented with a neural mass model, and all those neural masses are coupled together as defined by that individual's connectome. And the model generates a population activity for each node or region of interest, and that can be used to get functional outputs or features of interest. So in this case, we have empirical functional MRI data. And so we apply hemodynamic response function to get bold signals from our model, and we compute our functional feature of interest. In this case, I've used an example of functional connectivity. And we use the empirical data and compute that same feature, so functional connectivity, and we perform some parameter fitting to tune our models until they best capture that same feature. Right? So typically we also have data that speaks to an individual's cognitive function or behavior, and we can use those additional empirical data to determine whether our models offer some predictive value about that individual's cognition or behavior. So on top of the usual challenges that I think we all face as a community, so for example, whether we can reproduce our modeling data across different computing environments, across different platforms, for example, there are challenges that are unique to data-driven personalized models. And as you can see from our workflow, it's a long pipeline from raw empirical data to modeled outputs. And those data can pass through many hands. And often our teams are multidisciplinary, so data management and FAIR practices can vary across those different subdisciplines. And even before we get to the models, choices in empirical data processing can have profound effects on the empirical inputs to the model, and therefore could potentially affect model behavior. Those choices that impact empirical data can include things like which image processing software is used, which steps to include, the order in which those steps occur, and best practices in neuroimage processing are constantly changing. And so how we should process neuroimaging data is still at times contentious. So a related point is quality control of empirical data is really essential, and it's especially important with clinical data. So for example, with brain atrophy and our data sets of older subjects and subjects with neurodegeneration, it can be a real challenge to data processing, especially automated data processing. And this is something that's not often addressed. Like, quality control is not often prioritized in large data sets, which is understandable because it can be very time consuming. The size of a data set, if we were to consider both the empirical and model derivatives along the entire pipeline, can potentially be very large. And that can be challenging for making these models fully accessible and reusable. And finally, there are legal and ethical considerations around the protection of sensitive data. So for us, FAIR practices start with our data management plan. It's becoming more common for funders to ask for these plans in grant applications. So I know, for example, that both NIH as well as the Tri-Council agencies here in Canada have announced that they'll be implementing these in a couple of years. They'll expect grants to include data management plans. And it's something that we have found really useful in our work because of the size of our data sets, the long pipeline that we have, and our large team. And so the data management plan is a document that explicitly outlines things like data provenance, plans for documentation and metadata, our plans for storage, not only immediately, but also our long-term plans for preserving the data, the plans we have for sharing and reuse, individual responsibilities of each team member, resources we need, and whether we have them or if we still need to acquire them, and how we're meeting ethical and legal obligations. And so this is a document that we create as a team at the beginning of a project, and we review it as the project progresses, and we modify it as needed. Our FAIR practices in our pipeline continues with the empirical data processing. And what we've done is build a standardized processing pipeline for various MRI modalities to generate our model inputs. So it is based on the UK Biobank pipeline, which we chose because it was designed for extremely large data sets of older subjects. And we've added additional features to improve its robustness, and this includes the ability now to handle multiple MRI manufacturers, additional types of MRI acquisitions, and it has considerations now for special populations. So it not only handles aging data well, but it also handles images from individuals with neurodegenerative disease, and we are currently working on support for developmental data. And specifically for modeling, we've added the option to specify a user-defined parcellation in order to get the modeling inputs we need. And so the outputs are in BID's neuroimaging standard, and they can be used directly as inputs to the virtual brain. One of the best features of this pipeline is the detailed quality control reports that we've developed. And that allows for quality control checks in a fraction of the time. It includes a set of metrics that can be used to flag problematic outputs, and we're working on standardizing the annotations for these that accompany these reports so that we have from those annotations, a rich set of metadata about the empirical inputs to our models. And so if you're interested in more details on this pipeline, I encourage you to go check out our poster presentation this week. So now that we have those empirical inputs, we can take those into our modeling platform, the virtual brain. And the software for the virtual brain is available for Linux, Windows, and Mac operating systems, which you can download from our website at thevirtualbrain.org. It's an open source platform, so you can find our source code on GitHub. And there are a number of contributors to the project already, and we encourage everyone in the community to continue to contribute to it. There's also extensive documentation and training material, such as example code and videos, as well as a support forum, which you will also find on our website. And the virtual brain has a GUI interface, or it can be scripted. And so typically in our lab, we each have our own implementation of the virtual brain. It can be installed on a standalone machine, but more often we work with it on our cluster, or on our cluster, or a different high performance system. So everyone has their modeling pipeline scripted in Jupyter notebooks. They run their models, they get their data. And so generally I find that because FAIR practices were really baked into the design and implementation of the virtual brain, that it's easier to keep in line with FAIR principles in many ways at our modeling stage. And code and data are already in open formats, and users are generally sharing their code to repositories. Whether they can also share the data depends on our ethical obligations to those data, and that's been a challenge, for example, with patient data. But all that being said about our FAIR practices, this workflow is still in a siloed working environment. And because everyone has their own implementation, and as I've already said, it can be challenging to share certain kinds of data. So that brings us to one of the latest developments of the virtual brain, which is its implementation in the eBrains cloud system. A number of packages from the virtual brain suite are now available on eBrains, including the core simulator, an image processing pipeline, TVMe multiscale, which allows interfacing of the virtual brain with Nest, plus a number of other tools. So what this provides then is an infrastructure for sharing and processing large data sets in a collaborative environment. And so registered users can collaborate on the same code via Jupyter notebooks. And because it's a centralized resource for handling the empirical data and it has a modeling software, it allows for reproducible workflows. And it also has a security and encryption protocols to handle sensitive health data. So if you're interested in this cloud implementation of the virtual brain, I encourage you to check out the showcase in session 6 later this week, where there will be presentations on both the virtual brain cloud and the eBrains platforms. So where does that leave us, and what's remaining that we need to address? So first, we still don't have a standard for model outputs. standard file types, standard metadata. And that's a more general issue for the computational community. I know there's a lot of work that's already gone into this, including a proposal for a bid standard. So I think this is something that can be addressed and implemented very soon. The second issue has to do with data reuse and reproducibility. And assuming that we keep all of the modeling data, all of the model outputs, which isn't necessarily a trivial concern when we're talking about the sample sizes that we have, other than the modeling data, which empirical data derivatives are necessary for reuse and reproducibility? Is it enough to keep rich metadata describing how we arrived at empirical data inputs to the model? I don't know, and I don't think we really have a good answer for that at the moment. A third issue has to do with other neuroimaging modalities like MEG or EEG, where processing is less standardized, I would say, across the field, and perhaps there's less consensus there as to how best to process those types of data. And this is something we're currently working on, but it does remain a fairly large challenge for us. And finally, there will be new challenges as we start to integrate other non-brain data into our models, like genetics or metabolomics. And some of those challenges I think we can foresee, but I suspect that there will be many challenges to FAIR that we won't be able to predict until we go to implement those particular models. And so I'll end by saying that all this work is only possible because of a very large team of people. And that includes, of course, my colleagues at the Rotman Research Institute here in Toronto, but also our many collaborators on the teams led by Victor Jursa in Marseille, Petra Ritter in Berlin, and also our partners at CodeBox, who are the Virtual Brains software architects. And the partnership with CodeBox has been a really nice example of how one can partner with a for-profit company and still successfully have a fair and open science agenda. Thank you for joining me today, and I look forward to hearing from you during the discussion session.
Good morning, and thanks so much for inviting me to this panel. Today we'll discuss whether we have the tools and resources to develop fair, large-scale brain circuit models. My name is Salvador Dura-Bernal. I'm an assistant professor at the State University of New York, Downstate Health Sciences University. I will start by describing the modeling workflow that I follow, in particular with an example modeling the large-scale circuits of motor cortex. I will then describe the different tools and resources that I use for each step of the workflow, and I will outline some of the issues that we have and possible missing tools and opportunities for development of new resources. So I'll start with the example of one of the models that we built in the lab. Here I'm showing experimental data from the different cell types in motor cortex, organized by their projection target. And we are very lucky today because we have tons of experimental data. So it's important to find ways to combine this data to make sense of it and to build these models that can help us do this. So what we did is combine experimental data from over 40 papers from mouse motor cortex to build a detailed simulation of the motor cortex circuits. Here I'm showing what this model consists of. It reproduces a column, a cortical column in motor cortex of around 300 micrometers wide. It contains approximately 10,000 neurons and about 30 million synapses. So just to give you an idea of the detail of the model, we included the different cell types in cortex, different excitatory cell types, different inhibitory cell types. The laminar distribution of cells is based on experimental data. The density of neurons per layer is also based on several studies. Each of the single neuron models, so for each of the cell types, we build specific, very detailed models reproducing both the morphology. You can see here in the left, the original morphology reconstruction from experimental data and the simulation, as well as reproducing the physiology of each cell type. We also incorporated a lot of experimental data for connectivity. So we had connectivity at 3 different scales. We had information about the long-range inputs from different parts of cortex. We also had a lot of information about the local microcircuitry within the M1 cortical column. And we even had information about the distribution of synapses along the dendritic tree of different projections. So once we have the model built, what we did is try to validate the model. So we compared with experimental data of the firing rates of different populations and different layers obtained from different studies. And we showed that the model reproduces statistically the same firing rates per cell type and layer. And once the model was validated, we can use it to start testing different hypotheses and gaining insights into the actual biophysics and the circuit mechanisms of this motor cortex circuit. For example, we reproduce the activity different types of behavior when the mouse is quiet or not moving versus when it's moving in a treadmill. We also generated local field potential signals from the model and compared to the actual recorded LFPs in the mouse during the different behavioral states. So now I'm going to go over the different tools and resources that we used for each step of the workflow. These are the 5 steps I identified. This is by no means meant to be exhaustive, and the tools, of course, are not exhaustive at all. There are many other options that I will not mention here. So, of course, all these steps required, or ideally would require, FAIR principles, which means findable, accessible, interoperable, and reusable. So I will identify whether the different tools we used follow these principles or not, and what can be done to improve it. So in the first stage, the gather and preprocessing data, as I mentioned, this can be usually a very long stage, from 6 months to 1 year even. And the idea is to gather all the experimental papers that we require to extract the data from. So for that, of course, tools we use, basic tools is Google Scholar, which provides a great resource, but as well other modeling databases, as we can use existing models to extract data from them and use their references. So for example, ModelDB, Open Source Brain, the NeuroModel database, NeuroElectron, NeuroMorpho, which provide a lot of interesting data for single cells, the CRCNS databases, the Allen Brain Institute databases as well. And we also require tools to sort of combine and make sense of all these references and information we are downloading. So for example, we use Paperpile, which is this tool for gathering references. Google Drive and Google Sheets, for example, to make summaries of the data we obtain. And we use what we call electronic notebooks, just a way of recording what we're doing. And in our case, we use Virtual Studio Code with an extension called org mode. And finally, I wanted to mention this tool, Webplot Digitizer, which we've used quite a bit to extract or convert figures into data from papers. So in many cases, experimental papers do not provide all the raw data. And this is one of the issues which is related to FAIR principles. And this has the consequence of us having to use this kind of tools to extract data from figures, which is very tedious and time consuming. Once we have all this data, what we do is pre-process it so it can be used in the model. So of course, when we obtain data, they are in different formats. They have recorded using different techniques, different units. So we have to interpret it and combine it to make sense of it and extract some final pieces of data that we can input into the creation of our model. All this right now it's done sort of manually using Python. And at the same time, the provenance of the data used in the model, so the reference that we're using to obtain these data points, it's currently also stored in a manual way using Python or these electronic notebooks. So in my opinion, what is missing here? And of course, when I say what is missing, I'm referring to what am I missing? So I'm sure there are other tools I don't know about. And I will be very happy to hear from you if you have any feedback, any suggestions of tools that I should be using. So for me, an ideal tool for this step would be something where you automatically search and extract and preprocess the data from the literature. So I would like a tool where, in a perfect world, I ask a question such as, give me all the unitary connection EPSP amplitudes in millivolts between layer 5B PT cells and layer 2-3 PP cells in the mouse primary motor cortex during spontaneous activity. So of course, these are a lot of details, and it's hard to find this specific data in the vast literature. So this would be the ideal to have. And of course, to have it returned in a standardized format. I know there are potential existing tools that do something like this. I'm not sure about the stage at which they're in, but the NIX tool is something that I found which is related. The INCF knowledge space, I think, is also pointing in this line, and as well the Blue Brain Nexus. So the next step would be implementing the model once we have all this data. So for this stage, what we do is use as a backend simulator, we use the neuron simulation tool. And then as a network model building tool, we use NetPine. So NetPine is this package that we develop in the lab, which stands for networks using Python and Neuron. And of course, this was designed originally taking into account many of these FAIR principles. So we wanted to make it easier first to separate the actual low-level implementation code from the biological parameters of the network. So NetBine includes this high-level declarative language where you can define models focusing on just on the biological parameters and using things such as connectivity rules, which are very human readable. They're standardized, they're easier to modify in the future and to reuse by other users. The next stage would be the tuning and validating the model. So here we use several tools to automate this process. So of course, for modeling, we still use Neuron and NetBine. And for optimization, so parameter optimization, where we can set up a specific objective function that we want to reach. For example, comparing the fine rates of our model, different populations of the model to experimental data. We can use tools such as Inspired and Optuna, which provide these automated optimization methods, for example, evolutionary algorithms that run simulations and compare the outputs to experimental data and try to find the optimal parameters to reproduce the data. Both of these tools, Inspired and Optuna, are also built into NetBine, so it's easy to set up these kind of parameter explorations. So of course, the other big component here is the use of supercomputing or HPCs, high performance computing, as we need to run very large-scale simulations for these large-scale models and run many of them to optimize the parameters. So in our case, we use open resources such as the Neuroscience Gateway, Exceed supercomputers. We started using now eBrain's platform, and we also use Google Cloud Platform. So except Google Cloud, the other ones are free. So you just need to apply for these resources. And of course, also to mention that the optimization tools that we're using are also open source, and both Neuron and NetBine are as well open source. So this also follows FAIR principles. And the next step here, once we have the model already validated, and we want to start running simulated experiments and gaining insights and generating predictions. So in this case, we will still use the same modeling tools and supercomputing resources. And for tracking and managing the version simulations, I wanted to show here an example of how this looks in our electronic notebook. We have the different model versions. We make a note of the different features that were added in each case, and we make a reference to the GitHub change set corresponding to that. But of course, this is still very manual, and we have to do it for each of the versions sort of by hand. It's not integrated into the tool. And the same for all the simulations that we're running, all these parameter explorations. We have to make a note of each of them and explain what we're doing, summarize the results, and so on. So I grouped these three steps in the middle together to highlight some of the things that I think are missing. So the implementing the model step, tuning and validation, and the experiments and predictions. So one of the things that I think could be improved, and I'd be happy to hear any suggestions on tools in this respect, are the step of tuning model parameters to match experimental data, and the comparison to experimental data. I would like to see it more standardized and having more methods and formats which are standard in the field. And I know SciUnit and SciDash have made a big push in this respect, and they will be soon releasing their tool, which does this in a very systematic way. And I also know of the open source brain NWB Explorer. So NWB is this format for neurophysiological data. And so the NWB Explorer will be integrated in OSB and will actually allow you to compare simulation output data from NetBine, with the NetBine GUI, which is also going to be part of OSB, and compare it to experimental data sets that are available. So the other big component, which I have hinted at, is having more automated and integrated tools and standards for tracking and managing the model versions and simulations. So right now, as I was showing, it's mostly manual, and I would like to see some tools where you can actually store this information, the model versions, the list of changes in each model version, the provenance of parameters as well, the simulations that are run, the results associated with each simulation, all inside the same tool and in a more automated way. So I know of a couple of tools which have tried or are trying to move in that direction. One of them was the Sim Tracker, which unfortunately was implemented in MATLAB, so was not really open source. Open source brain as well has this concept of experiments, which you can see a snapshot here, screenshot in the right, where each simulated experiment is stored with some information. And as well in the NetBine GUI, we're moving towards this, where you can actually run these parameter explorations now in the cloud and then have a list of all the simulation results, and everything is sort of labeled automatically. Finally, the last step is sharing and dissemination. So here, I think it's crucial to have these standardized formats to share data and models. So of course, NetBind has its own, NeuroML and Sonata are trying to become standards for networks. As well, we are trying to move towards using NWB for the output simulation data. And there are several tools for sharing where you can share these models and the data from your simulations. ModelDB, of course, has a large repository of existing models, open source Brain as well, GitHub, which is becoming also a standard for modeling, so you can version your models and share them with the general public, and the eBrains platform. So in this final step, what do I think are things that could be improved or are missing? So as I mentioned before, again, standards and tools for the provenance of model parameters. So we currently, when we publish the paper, we sort of include some of them in the paper or in the code. I know, for example, the Allen Brain V1 model, they share the provenance of parameters in a very long PowerPoint, which was a very nice way of doing it, but it would be nice to have some more standardized methods. The other thing I think would be useful is to have standards for the high-level specification of networks. So for example, in NetBind, you can do this already with these connectivity rules in a declarative format, where the idea is that you have this sort of high-level definition of the network that can be used to generate the same network. And you don't need to have the 30 million synaptic connections defined one by one. So NeuroML also is developing this tool, this extension called NeuroML Lite, which has a similar high-level specification of networks. Next, I would say some of the other tools or resources that are missing is a way to run and visualize existing models. So a robust tool that is easy to use and is online, I don't think yet exists, but there are several approaches that are being developed in this sense. So for example, ModelDB, OSB, eBrains, and the NetBind GUI. Finally, I think it's important for the community to start adopting these standards and tools so that they actually become standards. So let me just finish with a screenshot from these 4 tools, which are providing this kind of online systems to run and visualize models. So here, top left, we have ModelDB, the right open source Brain, and the bottom eBrains and the NetBind GUI. So thank you so much.
Hello and welcome. In this talk, I'd like to take you through some thoughts related to structured validation processes in neural network simulations and analysis. To set the stage, I'd like to discuss some of the different scenarios where we are faced with the concept of validation in computational neuroscience. Then focusing on what you may consider as a classic model building cycle, where iterative improvement of models is constantly validated against experimentally available data, I'd like to highlight some of the tools currently developed to implement rigorous workflows for model validation. Within these workflows, I'd also like to briefly show the importance of validating analysis methods themselves. Now, in computational neuroscience, there's an increasing interest in the topic of validation, from model building to sophisticated analysis, as a means to expedite progress. There are many ongoing efforts to define the terminology and formal frameworks for validation in computational neuroscience. And I'd briefly like to point you to this classic picture of a validation cycle with the terminology chosen to match that of computational neuroscience. Here we consider a certain system of interest which is put into a mathematical model by analysis and modeling, and in turn is implemented as an executable model, which in turn is verified against the mathematical model to ensure that it's a faithful reproduction of the mathematics and correct in its implementation. The outputs of the executable model are in turn validated against observations taken from the system of interest. From this, we can then determine the amount of agreement and define whether this is acceptable. A very similar scenario, which we termed substantiation, is depicted in this picture, where again we have a system of interest which is put into a mathematical model, but here we now are dealing with 2 executable models, each verified in its own way. And here the question now arises in how far these 2 implementations of the original model agree or disagree. There are many aspects to take into consideration when talking about validation, whether to validate models against experiments or models versus models, whether to validate analyses, what is the degree of acceptable agreement, are we talking about validation or more calibration, and on which level are we observing validation phenomena. Here I'd like to consider network activity data as a special case of a model validation scenario, but I think many of the concepts I will be talking about will agree likewise. In the following, let me guide you through a typical validation and analysis process and the tools that are developed for this in the context of eBrains. We start with simulated activity data and experimental activity data, and the first thing we need to do is define a common data model that allows us to jointly analyze and compare these different inputs. For this, we use NEO, a common internal data representation for activity data and its individual relationships. The key concept here is not to have a common file format, but really I/O bridges to load data from different partially proprietary sources. In this way, we can take in data from experimental data, but also from simulated models. NEO itself is completely free from any semantics. These are completely delivered by the object hierarchy, by the structuring elements of NEO, and by annotations you can put onto the data. In the next step, we want to analyze data and extract meaningful statistical measures from the data we obtained. To this end, we're developing the electrophysiology analysis toolkit Elephant as a software library to provide a wide range of basic and advanced analysis tools for experiments and simulation, with a focus on coordinated activity in highly parallel neural data streams, and also considering multi-scale phenomena such as relating spikes and mesoscopic signals such as the LFP. An important aspect when designing such libraries is to validate against prior publications, because only such a validation ensures that indeed the statistical features you extract from data and use for further validation processes is in fact in line with the existing literature. To give you an example, I show here a scenario that occurred where we received a contribution for a reimplementation of an analysis method, in this case the unitary events method, which we then reproduced against the original publication from 1997. And of course, in the beginning, although in general the implementation agreed, in the details it did not. And this was not an effect due to lacking verification measures. The code itself was perfectly fine, but in fact it does matter on the implementation details on what you get out. So in the end, and after a lot of archaeology into the data, we were able to come up with an implementation that produces an exact match with the original implementation. And to make sure that it stays this way, these tests against existing publications are then manifested as unit tests into the library, making sure that all further validations have a solid footing. We may then continue to validation testing, which is covered by a whole range of validation libraries. Here I would like to focus on one of these libraries, focusing on network activity data and its statistical features. Other libraries which exist focus on either individual components such as individual neurons or their morphology, or individual systems such as maybe hippocampal data. However, the system of all of these validation libraries is quite similar. They are all based on the SciUnit network framework, which is essentially a way of specifying unit tests for science. Essentially, the structure of such libraries is that they preclude a certain number of capabilities. So in our case, this could be the capability to produce spikes, from which we derive certain tests you can perform on such capabilities. So for instance, calculating a firing rate. And from these generic tests, you can instantiate specific tests which have a certain data set in mind, certain parameters in mind, and a certain score to solidify the comparison into a single number in the end. You can then basically run the judge function to validate a certain model that has the named capabilities and end up with a test score. And a similar workflow will be possible for substantiation workflows, where you can essentially replace the experimental data by another model run. To show you how these validation libraries work in practice, let me come to an example of substantiation where we considered the so-called polychronization model, which was published by INCF in 2006. Now, for this model, both the original C code exists as well as a re-implementation using the NEST simulator, and we aimed to produce yet another re-implementation of the model using the Spinnaker hardware. And of course, our question during this development was, to what extent is the data from these individual simulation platforms comparable? In the end, we implemented these models such that they interact with the network unit library. And what you see here is on the left, a graphical representation of the output of the original C model, as opposed to 3 iterations of continuously improving the Spinnaker version of the model. Now, already by eye, you can see that going from iteration 1 to 3, increasingly the activity starts to match more and more that of the original C implementation. However, of course, we want to quantify this, and this is then done by, in this case, the network unit library, where you can see that measures such as the firing rate, the local measure of variation, and the cross-correlation coefficient continuously start to match that of the original C of the implementation as we improve the Spinnaker model bit by bit. You can see that here in the third iteration we still have some discrepancies in the cross correlations and the firing rates, but after enough working on the model, at some point you will end up with a quite comprehensive set of statistical measures which compare all kinds of aspects of the activity generated by the 2 implementations of the model. And all in all, we could say that we have reached quite good agreement between these 2 implementations. However, this is a very difficult concept since, of course, these 2 implementations will never match completely due to numerics, for instance. So we may ask, for instance, these 2 distributions of the firing rate, they are quite close, but are they close enough? Is this what we would call acceptable agreement? The question here lies really in what we are after. Now, you may know that the polychronization model was famous for producing certain spike patterns in the network activity data. So what we did is we analyzed the data now using a test based on the state method, which is a method to detect such spatiotemporal spike patterns, such as the one shown here in red, from the data. And what we observed was that although in all of these statistical measures that I've shown you before, we have quite good agreement, in fact, when we look at individual spike patterns, we observe a lot more in the Spinnaker implementation as opposed to the C implementation. And an educated guess is that maybe it's in fact this discrepancy of the firing rates which simply leads to an excess of patterns in the Spinnaker model as opposed to the C model. So in other words, if you're interested in patterns, maybe the small differences in the firing rate distributions do matter. In other words, acceptable agreement depends on what you're interested in. Finally, I'd just like to quickly point out that after going through all of these stages of analysis and validation, maybe you want to share your results with the world. And here I'd like to point you to the model validation framework developed by Shailesh Apokatan and Andrew Davison, also in the context of eBrains, where building on these individual validation test suites, you have a validation framework featuring a REST API plus graphical user interfaces in the eBrains collaboratory, which allow you to not only browse models, but also browse the corresponding validation tests, and likewise insert your own models and also insert your own validation test results that you have achieved for your model. This is, of course, then a resource that not only advocates and exposes the models you're creating, but directly tells users to what degree they can reproduce certain features of our own world. So in summary, I hope that I've convinced you that validation is really a very multifaceted theme for computational neuroscience. We are dealing with model validation and model substantiation. We're dealing with validation of analysis codes, and also, what I didn't touch upon here, cross-validation of complete analysis pipelines. Also, I hope you've seen that there are emerging tools that cover data acquisition, simulation, analysis, and validation workflows. And hopefully these tools will all grow together to form an ever-expanding ecosystem of tools to cover all aspects of validation and provide a faster growth of science altogether. I'd like to point to a workshop and infrastructure training held in May regarding these model validation aspects, organized by Andrew Davison in Paris. And with this, I'd like to thank you for your attention and hope for a discussion. Thank you.
Great. Thank you so much. Those talks were really great. We welcome questions from audience out there now, and feel free to post those, Umbrella, and we'll try to incorporate them into our discussion here, our roundtable discussion. So I want to thank you guys. Those talks were so great. I knew they would be, and I enjoyed them very much. Another thing that was fantastic was that you guys all talked a bit about what some of the current challenges are in FAIR, or, around all these different pipelines that we use. And in particular, some of the technical challenges and what you see as some of the future challenges. Maybe we could start by talking a bit about what you think are some of the greatest remaining social challenges. So I know when I first started working on reproducibility in computational neuroscience, it really was the biggest challenge. Nobody was really very interested in what we were doing. And there's such a huge change now. And I think the community sees the value in FAIR approaches, but we still have a ways to go. So you guys feel free to unmute and just chime in. What do you think are some of the greatest remaining social challenges?
I'll start.
Okay.
Yeah, I think adoption hesitancy is still an issue, and there are legitimate reasons to the hesitancy in sharing openly data and code, for example. And one I mentioned is that something we can overcome with technical solutions, which is kind of the ethics around sharing private health data for our personalized models. But another challenge is kind of the barriers that different people face and the consequences they may face if they were to consider sharing. And those are kind of systemic or structural barriers for early career researchers or trainees, for example, and some of the hesitancy around there. And I think those require a broader discussion and broader systemic changes. And I'm hopeful that with kind of reproducibility, as you say, being more readily adopted, that even at the training phase, it's already part of the culture. And that's something that will change over the generations.
Yeah, and
Salvador?
just to add to that, I think that following FAIR approaches takes more resources and more time that nobody has in research. So it's something that it seems we all want to do, but if you have to take something out of the equation, that is something that often is taken out. So I think maybe more enforcing from the grants points of view. So if the grants actually require these things when you're submitting them, it would force more people to do it. Even from journals as well, which some of them are doing, but some are not, and to not such a bigger extent of all the whole FAIR principles. And then it would trickle down from PIs and forcing the lab members as well to do it, and so on. And the other big thing, I think, is more dissemination of all the tools that are available and standards and so on. Even us who are, I think, quite involved with the field are missing a lot of those. And I've just realized, just listening at your talks and the whole INCF conference, there's a lot of things I don't really know about. So yeah, more dissemination in general, I think, would be useful.
Miguel, do you have anything to add to that?
Yeah, I fully agree. I have one more point, which I also find rather often, and which I find is to a degree maybe a social problem. So I would say that sort of working on these solutions to more fair data processing is maybe in physics terms, you would call it a dissipative system, at least in the beginning, in terms of that energy is wasted very often. So what I see often is that students, like PhD students or so, come to the lab and they're very enthusiastic and they really want to also work on these issues. So they want to develop nice solutions and so on. And so they do, because it's fun to do something new. And typically there's no PI to stop them or to tell them, that's great, but why not build on what's already there? And so basically they work for a year and then they go to the first conference and they see, oh, there's already stuff happening. And then maybe they have a great idea, but it's completely standalone. And I think a social thing I would see as a big challenge is to get into a mode to much earlier address people who start developing things and give them the chance to see what's out there and also to make it understandable what's out there. So often I think people just don't understand the tools that are out there because they are described at so high level terms that it's difficult to understand. So I think it's a communication change, I would say, is very important.
Yeah, I want to just comment really briefly on something that Kelly said. She was talking about time, or Salvador said maybe talking about time, and we don't have a lot of time. None of us have a lot of time. The way I've always viewed things like standardized descriptions and validation is that all of these aspects of FAIR need to be invisible to the standard modeling user. As Kelly said, a lot of these things are built into the virtual brain. Salvador obviously is doing similar things with NetPine, trying to, build these FAIR structures into the software. And I think that's what's really important. We don't want this to take more time. We want people to be able to push a button and the standardized version is there that they can share with the world, right? And so that's why I think, talking about this in the context of neuroinformatics at the INCF is so important, because all the tool makers, we're the people who need to be thinking about these FAIR approaches. We have a question from Anita Bandrowski. In your opinion, what are the most widespread and accepted standards in the field of computational neuroscience? And what are some of the challenges that you see with adoption of these standards? Anybody want to go first? Maybe Salvador?
Sure. I mean, from my perspective, what I deal with, I would say in terms of biophysically detailed large-scale networks, I think NeurML and Sonata are both gaining terrain. But it's true that adoption is very low for what it could be. And there's a big effort from the sort of developers of the standards and some of the developers of the tools actually making the standards available in their tools or easy to use. But it's true that it's most users who develop models don't make an extra effort to convert to the standard unless it's very easy or given by the tool. And I think the issue is the reward you get from it. As I was saying before, there's nothing really you gain extra once you publish the paper. Why would you spend time converting to a standard? Of course, we see the advantages because it makes it usable for many other people in the future and to modify and so on. But again, it's the limitation of resources and time, I think.
Anybody have anything to add to that? Yeah, Mikhail?
Yeah, I think I would agree that I think standards which are closely coupled to specific tools are probably the most widespread in a way, and probably also most adopted. But I think what's really difficult is this transition then between tools. So if you take standards for describing models, for instance, of course there are attempts, but of course you always have this gap that you also have, for instance, with data formats and other things. You always have very specific solutions which work well with a tool, but how do you build this interface to other tools? And then we come to the whole question of standards which would require some sort of more widespread ontology or some at least vocabulary in the first case, where I think we need more people dedicated to working on this and finding a common language that also allows us to standardize on a broader scale.
Okay, I want to ask one more kind of general question, and then there's a specific question for Mikhail, so we'll come back to that in a minute. This was alluded to a little bit before when we were talking about social challenges, but what challenges do you think should be made to at the level of graduate training, early career training in computational neuroscience that might better support FAIR principles? Salvador?
Sure. Yeah, I think they should be included as an integral part of training, not as an optional thing. But when you teach them from the beginning, this is how you have to do it, and these are the tools you have, and also highlighting the issues and problems when you don't do it. So these are the advantages when you have a standard tool, and the next postdoc student will be able to use it in a very easy way, and so on. And if you don't, this is the mess you have, and this is how difficult it gets. So.
And how do you think we could best, help trainers? What do we need to help trainers bring these tools to students or bring this information to students?
Yeah, it's. I guess it's also a bit of a matter of funding as well to get opportunities to develop this kind of workshops, courses, tools in conferences, and so on, where we have the time and the resources to present the tools to the actual researchers and the students. And again, it's a limitation of time because we want to do the research and not the dissemination. So agencies, maybe funding agencies, should get more involved in forcing this kind of dissemination courses.
Kelly, did you have something to add?
Yeah, I was going to say that, adoption within a lab is the easiest way to train the next generation of students. So by not just example, but by expectation that at the beginning, at the outset, when trainees join, that we are going to practice FAIR principles, and presenting it in a way where it's obvious what the positive side, other than that, it's great that we're producing reproducible science, but the opportunities that trainees get, for example, once they share their work for networking and other things that are actually very positive outcomes, I think need to be made clear to trainees and their participation in FAIR.
Anybody else? Mikhail?
Yeah, I think I would add two small comments to this. So I think one thing which may really help is, now that we're in a situation that more and more data actually is becoming FAIR, I think it would be really helpful to include data that's online and FAIR data into the training curricula, so that people actually see the benefit of having data open and accessible, so that it's really that you experience this benefit, because I think that's something that's often come up. And maybe the other thing is that I recently was in a discussion which I thought was really nice, and then after some time there was again this comment that, oh well, but I don't want to share my data, and this typical stuff. So I think it's important to train the young generation, no question. This is the key. But we shouldn't forget about also teaching maybe the not so young PhD students anymore, but continue teaching what would be the advantages also on the PI level and so on, so that these prejudices get less less in the way.
Yeah, on that note, I guess since the Allen Institute started and from the beginning, they had these FAIR principles for their data, sharing their data. I think they set a really good example to the community about how you can make everything open from the beginning. You can publish it. Everybody else can use it. They'll publish about different things, right? And it's just better for everyone. So I just always have thought that is great to have that example. And there are others as well out there. As the community moves more toward open data and models, I think it helps everyone. All right, let's address a couple of these very specific questions. So, Mikkel, you can see this question in the chat. To which extent does the unit test framework, how is it applicable to the validation, quality control of anatomical segmentation and voxel-based results? So maybe speak a bit about the validation framework for anatomy.
Yeah, so so in general, the idea, and I have to say this, it's not my idea, but the SciUnit framework exists now for quite some time, but I think it's an extremely appealing idea to basically have a framework and a formalization of these validation type processes, where basically the language and the processes are standardized, but of course then the adaptation to a specific field needs to come from the communities, which is why there are so many attempts now to build different types of validation libraries for all kinds of models, and these are on all different levels. So some are like what I presented as on the level of activity data. Some can be more on the level of maybe a specific region, so maybe a hippo unit as a hippocampus type validation set and so on. So I'm not an expert on any anatomical data, but I'm sure that one could also have corresponding types of modules which then expose certain capabilities, certain test strategies for comparing here models versus experiment versus experiment, model versus model, etc. So I think in general, the interesting thing is that this is again a thing where I think computational neuroscience needs to slowly build up a vocabulary of what we are talking about. So what is validation as opposed to, for instance, calibration? Yeah. So and so on. Yeah. And so we talk in a common language. And I think software tools can be a great way to do this.
Yeah, I'll just add that if Greg or anyone else in the audience is interested in seeing a list of these repositories that are out there already for different areas for validation, just get in touch with me or Mikkel and we can point you towards those. There's also a specific question for Kelly. Kelly, it's kind of long, so I think I'll let you just, say the parts of the question and address it yourself.
Sure. This question is from Marianne about data management plans and how, often we think about them as just grant administrative supplements. But I like this comment here about how the library community is and want to make the machine actionable. And that's great, because especially in the context of what we're talking about here in INCF and how INCF can maybe support better adoption, I guess, of data management plans, I think is maybe what the question is. INCF provides some really great training materials. So there's a space there, I think, for providing a good description of what a data management plan is, what the lifelong kind of project lifespan of a data management and how plan changes, for example, and maybe some templates and examples for common types of neuroscience data would be a really I guess, nice collection for adoption of data management plans.
I think, Kelly, you gave this great example in your talk, and I was thinking, oh, I'd love to see all the details of that plan. So I think that's a great idea to have these templates available to people, because if they aren't here listening to this talk, they won't see your plan, right? So that's a really good idea. Okay, so there's also a very specific question from Tom Gillespie for Salvador. Salvador, why don't you take that question next?
Sure. So do you also use the Emacs version of org mode? So we started using the Emacs, and that's my previous PI used that one, and he sort of forced it onto everyone. And I got out of it because I found it really hard to use the Emacs, but other people love it. So do you have an onboarding for that? Kind of. The VS Code one is not really hard to use. I only use mainly the feature where you have many hierarchical levels, which I think is really useful for note-taking in research. And I find it much easier to use in VS Code than Emacs. And I really haven't found any other sort of text-based tool where you can do this hierarchical organization in an easy way. But yeah, the Emacs VS Code is a big fight between it.
Okay, great. All right, let's move on to another kind of broad question. And this might be mostly for Kelly, but how do you think as a community we should approach the ethical challenges that are related to FAIR in computational neuroscience? So, I mean, obviously with personalized data, it is a big challenge, but it's something that the wider community is addressing, the data community. So maybe you can tell us a few more details about that and what might reach into, all aspects of computational neuroscience as we move towards more personalized approaches.
Right. I think this is where kind of keeping in mind the difference between FAIR approaches and open science. Right? We're not going to be able to meet ethical requirements if we're going to make everything open, and that's the balance, right? And so, the example that I've given in eBrains and the virtual brain in the cloud is one way that we can stick to FAIR, but not necessarily have everything open, because it requires a number of security features and people have to be registered, and there are certain aspects of the data that are anonymized and secure that way. And so I think that would be kind of keeping FAIR principles separate in our heads from open science is one way to continue to address those ethical challenges.
Anyone have anything to add? I think there's a session maybe later on about ethical challenges. And so maybe we can all learn from that other session in the INCF assembly, because I think it's going to be becoming more and more of an issue as we as we start doing more personalized models and using models in medicine more and more. Okay, great. Heather's gonna tell us about this session on neuroethics, so we can all be sure we attend that. Okay, so maybe we can move on to another broader question. And this is stretching us a little bit, but what do you think will be the biggest challenges facing our community related to FAIR, in the far future? You guys can also expand a bit on some of the things that you brought up in your talks, which I think were great in terms of technical challenges. So, I think we're starting to reach some of the lower level challenges in FAIR. We have these beginnings across these themes across FAIR, but we still have a long way to go. So what do you see are some of the major technical challenges, and what do you see they might be even stronger challenges in the far future? Salvador is ready to start.
I'll give it a go. So something I mentioned briefly in the talk is the ability to, for example, reproduce models once you build them. And this is starting to become an issue because they are becoming much larger models, much more detailed. And of course, in the future, if we start building models of this level of detail of large areas of the brain, it's going to require like massive amounts of resources, both to run them and to send data back and forward. And the same goes for the experimental data that you require to build the models, to validate them. So I think it's an issue how individual users, individual labs are going to manage these massive resources to be able to move models, run models, and data back and forward.
Anyone else?
I was going to add to that by saying, often we think about the future of technology as kind of having some infinite resource for data storage and data sharing. And I don't think we often think about that, it's not infinite. Right? And there are significant challenges to not only keeping kind of like the type of storage and media, consistent or at least accessible over long periods of time, but in kind of this big data world, there's a serious physical impact of the hardware that we use in these storage farms or these server farms in terms of, for example, climate change and whatnot, and all these that we're going to hit eventually. So, I think we got to start thinking about ways to minimizing that impact in terms of what fair principles or kind of like metadata or data that we need versus to reproduce versus what is actually just extra.
Mikhail?
Yeah, I could also imagine one of the challenges. So thinking back again some time, I think when we talked about sharing data and making data public and so on, was not only the fear of making your data and models and whatever public, but also the fear that once the data is out there and the models are out there, they are so complex, whether this would not lead to a lot of bad results, because people just would not be able to understand the data or the model at this level of detail which they need. And I think to a certain degree, I think we've gone a long way in terms of it's much better now already. And I think we are coming to processes where I can get data and understand it reasonably well. For me, the next question is that we are also talking about data mining and automated agglomeration of data across very heterogeneous data sets. And I think this question now starts to repeat again a little bit. So what happens if I really do this automated harvesting of data and so on? Can we really get our descriptions of models and data to a stage where we can then also trust these results? Can we really put all of this implicit information into the descriptions of the data so that we can actually make large-scale use of these data things? I think that's for me like one of the questions I would have for the future.
I think that's a very important point, just to note that right now we do a lot of it sort of by hand because the models are still manageable, but as they grow, they're not really going to be manageable by individual humans. So we're going to have to move to more fully automated approaches in terms of optimizing parameters and understanding the results and so on. So that's going to be a tough problem.
Yeah, I know even resources about single cells like Neuroelectro, right? So they did data mining of the literature to fill in their database with experimental observations about single cells. And, as far as I know, it had to be heavily curated. And so certainly when you get to the point where you're talking about networks and, large scale data, anything around automation just seems like a huge challenge. So Salvador and Kelly in particular, you spoke in your talks about, relatively large scale models. So could you say a little bit more about, the specifics of how you would share those kinds of models with the community if somebody, if you wanted somebody to be able to rerun your rerun your model. What can you do now, and what would you like to be able to do in the future?
Sure, I can start. And right now, our models can be shared, for example, on a repository in GitHub. But to reproduce them, we also need to share the model inputs, which are the individual data sets, which is quite a bit. And we have resources now to share that, for example, in OpenNeuro and Zenodo. But, and also keeping in mind with some of the discussion today already, what we would like to see is more standardized approaches to the sharing, but also the parameter fitting, the model validation, all those stages are not as fair as or as they can be. So I think those are some challenges that we face, I would say almost immediately, that I'd like to see addressed.
Yeah, so yeah, right now we do share the model code in NetPine code, which is sort of standardized and can be exported to Sonata and NeuroML. So the model definition itself is standard in that way, and we share it on GitHub, ModelDB, and Open Source Brain. But it's true that we don't really provide a way to fully reproduce the results identically, because that depends a lot on the platform. And we have seen that ourselves, like even although NetPine is built to try to reproduce on different platforms and different number of cores, there are sometimes small issues if you go to different supercomputers and this kind of thing, where you might not find exactly the same result, which of course is an issue. And the other big issue, I think, is this idea of provenance that I was talking about as well, and the actual sort of model building process, where we have gone through many different stages and adding many different features. And I think it would be really nice to have both the provenance of the experimental data and the model building stages in a much more standardized way. So it's easier for people to understand what happened, where the data comes from, and make changes in the future and so on.
So I think both of you mentioned, this validation stage and having better automation, transparency in that stage, which is essentially what Mikhail was talking about in his talk, which is great. So all your talks fit quite nicely together. So maybe Mikhail could comment a little bit about, some of the challenges around validation and how it could be made maybe a little bit easier for the average user. I guess essentially, eventually it would be nice to have that built into these platforms, built into sort of some sort of, as Salvador said, building these standardized sort of tests into NetPine, for example, building this into workflows for the virtual brain. But Mikhail, maybe you could say a little bit now about, sort of now, what are the challenges until that happens?
So I use, yeah, I guess okay, yeah, sorry, there was a hiccup. So for me, one of the challenges I see currently is that the data and the models are still very disparate. So both in terms of their presentation, but also in terms of the metadata that they encompass. So for experimental data, and now I'm taking the example of activity-like data, so maybe electrophysiology recordings from networks and stuff like this. I think currently there's quite a bit of effort to try to dig a bit deeper in terms of metadata, describing not only this was the author and this was the system, but really a bit more deep, like what's the geometry of electrodes and all this kind of stuff. Of course, we are not there yet, and this is a slow process. But on the other hand, I have the feeling that for models, we should start a process that that goes alongside with these efforts, so that once this is more progressed, that this automated connection between these two worlds will be possible, because otherwise we have very nicely annotated experimental data, but it will be unclear how to relate this to experimental data. So for instance, assuming, now again, stupid example, but assuming you have, for instance, a 10 by 10 electrode array recorded in some area, and the metadata tells you what's the spacing, what's the depth, maybe even some coordinates and stuff like this. And on the other hand, maybe you have some sort of model that's, I don't know, it has 2 neuron types and has, I don't know, 1 million neurons in the same area and so on. So how do you describe these two systems so that you can make the match? Like you would have to subsample certain neurons from one system and relate them to the other and these kind of things. So if you really want to automate this process and make it easy for the user, I think that a lot of key issues are, again, in the metadata, I feel.
Yeah, I agree completely. I think we discussed with Marianne Martin a while back. There's like a big missing link between the experimental data and when you started building the model. He was saying the disparate representations and so on. And it's hard for modelers to actually find the data they're looking for, even though there's so much data out there. So I think there's a big space to improve the workflow there.
Yeah, I agree. I think that, well, this is something that the model description standards community is definitely paying attention to. You know, how to associate metadata with models. Another thing is, if you describe models in a standardized way, you know, then you can query the data about the model itself. I mean, you know how to look at the standard and get the information that you need about you know, how you can do validation, for example, against experimental data. So just using standards, you know, gets you a long way towards that. Okay, so any final words? I just want to thank all of you. The talks were fantastic. I really enjoyed hearing everything that you had to say and enjoyed this discussion. I think you brought up a lot of interesting points, and I'm looking forward to going back over the slides and looking at some of the challenges that you brought up for the community to try to address in the future. So thank you very much. And I guess we'll just stop a couple minutes early and everyone can get ready for the next session.